Uh, Now today we will discuss on urea cycle. This is in the syllabus of semester 4 of UG2. We know that after digestion of the protein, the final end product is the amino acid. Amino acid contains amino group. This amino group with carbon dioxide form urea. Urea is the main nitrogenous waste product. Among all nitrogenous waste product, it constitutes near about 90 to 92%. This is very toxic, so that should be eliminated or excreted from our body in urine. So now we will know how urea form. Formation of urea actually takes place in liver, and from there it is excreted, come into kidney and excreted in urine. In metabolic process, the carboxyl phosphate synthetase enzyme, carboxyl phosphate synthetase enzyme convert carbon dioxide and ammonia into carboxyl phosphate, and this enzyme is found in mitochondria. So the first step of urea formation is the carboxyl phosphate synthesis. After the formation of carboxyl phosphate synthesis, the enzyme ornithine turns carbamylase, converts citrulline. That means citrulline formed by the combination of ornithine and carboxyl phosphate, and that takes place in mitochondria. So this cycle is also known as ornithine cycle. The urea cycle is also known as ornithine cycle because citrulline is producing on with the help of ornithine. Now see, citrulline then converts into arginosuccinate with the help of aspartic acid or aspartate, and this takes place with the help of arginosuccinate synthase enzyme. At that time, this process utilizes ATP, which convert into adenine monophosphate and pyrophosphate. So the second step of urea formation, first step was what? Carboxyl phosphate synthesis. Second step what? Citrulline synthesis. And third is the arginosuccinate synthesis. Then after arginosuccinate cleave into arginine and fumaric acid with the help of the enzyme arginosuccinase, which is found in the cytosol. So this cleavage of arginosuccinate, arginine and fumaric help the synthesis of urea. That means here arginine used as precursor for urea. Actually, urea contains two amino acids, one coming from this amino group of the amino acid, and other amino group comes from this compound arginosuccinate. So here you see that I have written the formula NH2 CO NH2, two amino group are there. So when arginosuccinate gives arginine and fumarate, fumarate tends towards the Krebs cycle, and now arginine converts into ornithine with the help of the enzyme arginase. At that time, urea synthesis takes place. That means this is the final product of urea. So ornithine again tends to the same process with the help of carbonyl phosphate, and same in mitochondria gives rise to cytolin. So during the conversion of that process which takes place in the liver, hepatic cells, first of all, the conversion of these two CO2 and NH4 ammonium ion, whenever going to form carbonyl phosphate with the help of carbonyl phosphate synthetase enzyme, at that time it uses two ATP molecule which converts into two ATP and inorganic phosphate with the help of the NAG and acetyl glutamate. That is important because that makes the enzyme active. That's why we apply the N-acetyl glutamate, NAG. So this is the urea cycle which takes place in liver cell. Few steps takes place in mitochondria, and then most of the process takes place in the cytosol. Here we are getting two main components, ornithine and cytolin. They are very important amino acid, fresh amino acid, and they are not synthesized by the process of genetic code because their synthesis does not contain DNA or messenger RNA. So here these are two important amino acids. First of all, where we are getting cytolin with the help of ornithine, because ornithine and these two combine cytolin. That's why their cycle, that means urea cycle, also known as ornithine cycle. So this is overall of the process of urea, and the synthesis of urea is very important because it is nitrogen waste product that should have to remove from our body. And from where we are getting on? Now the first initial steps is the amino acid, which has amino group. The nitrogen containing this amino group, when detached from the amino acid, that converts into urea. The first synthetic process, and that should be eliminated from the body, otherwise the metabolic process which is going on inside of the body all will be toxic. So that toxic matter should be eliminated from the body in urine, and the formation of urea, you know where it takes place? Liver. Very good. It takes place in liver, and after that it is excreted, passes into the kidney and then excreted in urine. Understand? This is about urea cycle. Okay? Thank you.